Dear student, welcome to my YouTube channel. In today's lecture, we are going to discuss a very important topic, that is centromere, which is a part of chromosome. In this lecture, we will get to know about the structure of the centromere, as well as its important functions. So we will first define what is a centromere. A centromere, we all know, can be defined as the compressed region or part of the elongated chromosome. It is specialized DNA sequence in the chromosome that links or hold together the pair of sister chromatids. The centromere are also involved in separating the chromosome into short arm. The short arm of the chromosome is denoted by P and long arm is denoted by Q. So centromere is the region, uh, actually is the region specialized DNA and protein. Certain proteins are also there, and yet together hold, that is sister chromatids. The centromere is defined by specific DNA sequence plus proteins. There are certain special proteins as well that are present in the centromere region. The centromere is the point of attachment of the kinetochore, a complex, that is multi-protein structure to which microtubules of the microspindle being anchored. So centromere is actually involved in the separation of chromatid during the cell division. Structure of centromere is best studied in yeast, that is Saccharomyces cerevisiae. So here, that is a structure of that is we can say, here is the structure of the centromere. You can see this one is that is you can see from this figure, this is the chromatid region, this is the chromatid region, this is the kinetochore, that is the centromere region, which is also known as primary constriction. And here the microtubule, they are anchoring to the kinetochore region. These two are the sister chromatid, this one and this one. I can show you. This one is first sister chromatid, another sister chromatid, and their microtubules are anchoring towards the that is kinetochore region. This is the centromeric, that is chromatid, inner plate, that is then outer plate, and the microtubule region associating with these kinetochore regions. Now, broadly speaking, there are two main type of centromere. One is called as point centromere, regional centromere. Point centromere means it is having some only few, that is base pairs of nucleotides, nitrogenous bases. They constitute your centromere. That is called as point centromere. Whereas regional centromere, that means long chain, that is long sequence consisting of a number of reasons. We will study later on in this lecture about that is yeast centromere, which is having three main elements. We will discuss this later on. So here we will discuss these are the centromere where the mitotic spindle fibers are that is attracted to specific regions of the DNA. In this case, the cell protein bind to these particular DNA sequence and form the foundation for the binding of the mitotic spindle fibers. So those regions where only these specific regions are consisting of only few base pair, that is called as point centromere. And regional centromere, that is humans and most eukaryotic cells have regional centers. We all know mostly eukaryotics have a regional centromere, whereas prokaryotics, they are having the point centromere consisting of only few base pair. Then depending upon where centromere located, we have different that is chromosome, that is metacentric centromere are located near the chromosome center, that is that is two sister chromatids, they are of equal length, and centromere is located in between them, that is called as metacentric. Then submetacentric, that means they are non-centrically located, so that one arm is longer and other is the shorter one. So we are having the P and Q, that is Q long arm and P, that is short arm. Then acrocentric chromosome are located near the end of the chromosome, almost not, it is not terminal, but near the terminal, that is called as acrocentric. Then we are having the telocentric centromere. These are found at the end of the telomere region. These are again different type of chromosome depending upon the location of centromere. Then we have different proteins that is associated with centromere. These proteins are designated by a number of that is nomenclature, but we have specific, we will talk here only about certain specific that is protein antigens, that is A, CNP, that is centromeric proteins A, B, and C. These are three main centromeric protein antigen localized to centromere throughout the cell cycle. CENP-A and CENP-C are contained within the inner plate. Here in the last diagram we have seen there are two plates. One is called as inner plate and another is called as outer plate, and in between there lies the microtubular region. So centromere protein A and centromere protein C, they are contained within the inner plate, and centromere protein B, that is within the centromere heterochromatin. So these are having molecular mass of different ranges. For example, CENPA, that is having 17 kilodalton histone H3 or later protein, which is required for the assembly of the centromere and kinetochore components. Centromere is the part of the, that is the chromosome, and kinetochore, that is microtubular regions. CNPB binds to a 17 base pair motif of the CNPB box sequence that is found in the subset of centromere specific human alpha satellite DNA. Then CNPC is a highly basic, that is 140 kilodalton protein, which is binding with DNA binding and self associating properties, and is essential for chromosome segregation. That is, this protein involved in the chromosome segregation. That is, it is having the largest function of all, that is segregation of chromatids during the cell division. Then we will discuss about different in earlier slide I have discussed that centromere region is in a regional centromeres, it is consisting of different elements. Here the centromere of unicellular Saccharomyces or baking yeast are referred to as point centromere, which is approximately consisting of, that is 125 base pair. Directly centromere assembly and position, this 125 base pair is encompassing three basic elements, that is CDE1, that is called as cell cyclic dependent your proteins, that is elements, cell cycle dependent elements 1, 2, and third. TC, that is cell cyclic dependent element 1, it consisting of 9 base pair, that is conserved with minor differences. It is conserved in almost many organisms, that is consisting of 9 base pair, and this 9 base pair is consisting of nucleotide, that is thymine. This is the nucleotide sequence, that is thymine, cytosine, adenine, cytosine, adenine, thymine, guanine, adenine, then thymine, that is TCA, CAT, GAT. Then second element, that is cell cyclic dependent element second, it is consisting of TAA, TA, TA, that is, it is about 90%, that is 90% consisting of only this elements. 90% of the centromere in case of yeast is consisting of, that is CDE second, only 10% constituting your CDE1 and CDE third. It is 80 rich, that is adenine, thymine rich tandem repeats. Then cell cyclic dependent element, that is third, that is consisting of again 11 base pair sequence, which is TGA, triple T, CC, G, AA. Then there are also specific proteins which is called as CENH3. CN stand for centromere and H3 is a histone protein. It is also associated with the centromere region. Over 100 kinetochore protein form the point centromere on each that is chromosome, creating an attachment site for a single microtubule. That is over 100 kinetochore protein, they involved in the attachment of single kinetochore to the centromere region. In contrasting to the budding yeast point centromere, the centromere of larger eukaryotic are defined as regional centromere that span over more than several kilobases in size. So regional center in case of your eukaryotic cell is consisting of large sequences which are ranging near about kilobases, whereas in case of prokaryotic they are having the point centromere having only few base pairs. Then functions. About functions, the centromere are the point of attachment. That is, they are actually the point of attachment. We have discussed that centromere and the kinetochore, they associate with each other to form the that is segregation, to give rise to the segregation during the cell division. These centromeres are the point of attachment of the kinetochore. Kinetochore are the part of the microtubules, and they centromere are the point of the attachment of kinetochore. These are the reason where kinetochore will anchor during the cell division. The main function include the attachment of sister chromatid, and it is the site for attachment of spindle fiber as well. Then second function is centromere help in the proper alignment and segregation of the chromosome during the process of cell division. That is, these centromeres actually helps in the proper alignment as well, and as well as segregation of chromosome during the process of cell division in eukaryotic cells. Then centromeres play an important part in the production of a new cell. How? Actually the centromere are actually helping in the segregation of chromosome, so they play a very important role in the formation of new cell. When the chromosome are copied during the replication, these centromeres served as binding site for the two replicated chromosomes, which are known as sister chromatids. So before replication, that is, chromosome is consisting of only two arms, one is P and other is Q. But after replication, each chromatid is having a sister chromatid as well. So these sister chromatid attached to the centromere region, and ultimately these sister chromatids separate during the cell division. Centromere are also involved in the sister chromatid adhesion and separation and chromosome movement as well. Then microtubule attachment as well as the establishment of the heterochromatin and mitotic checkpoint control. These centromere helps in large number of functions. Centromere also function as sophisticated signal processing centers which are involved in the regulation of cell cycle progression, where cell that means centromere actually have to decide, as well as we have discussed about the CDE elements. These CDE elements play a very important role in the regulation of the cell cycle progression. So this is all about the centromere, its structure and its function. In the next lecture, we will discuss a very important part of the chromosome, another very important part of the chromosome, that is the telomere region, which are actually the end portion of the chromosome, and how these end portions are playing a very important role in the chromosome, as well as how they can be protected by mechanism, by replication. uh, we will discuss that in the next lecture. This is all about this lecture. So thank you very much.